Welcome back. We will continue today about gametogenesis, and in the last class we discussed about the spermatogenesis, and today we will discuss about the female gamete, that is ova, formation of ova, and what are the process involved in the formation of ova from starting cell, which is called oogonial cell. Before going exactly to the oogenesis, that is the formation of ova, let's try to understand one thing which was left undiscussed in the last class, and that is called the difference in between two terminologies. One is called spermiogenesis and one is called spermiation. So, spermatogenesis as a total, we discussed about the process where the spermatogonial cell. So, this is a kind of a mother cell, and this mother cell, which we are calling gonial, and in case of sperm we are calling it spermatogonial cell, and this spermatogonial cell is doing all the processes which we already covered in the last class, going to form a spermatid. So, this is spermatid. So, spermatids are nothing but the last product of the whole cell division during the formation of a sperm. But spermatids are going to convert into a motile form of the sperm, right? The actual developed form of the sperm by a process called spermiogenesis. So, what is there is called spermiogenesis, is nothing but the conversion of spermatid into something which is flagellar in nature, and that is called spermatozoa, which is a motile sperm. So, there is a process of differentiation. The cells are same, but there is a differentiation. Now, one has to remember we are talking about a division which is reductional in nature. So, we are talking about a meiotic division, but here the chromosome number is 2n, and here what we have is n because there is a reduction. So, from one cell we are getting 4 different sperms. If you see the actual chromosomal structure in the whole diploid cell, what is there is what we call 22 pairs of autosomes and then 1 pair of sex chromosome. This is in case of male it is XY, and in case of female it is XX. So, this is what is defining the sex of an individual, and basically what is there, we have 23 pairs of chromosomes. So, here we have 23 pairs of chromosomes, and here we have only 23 chromosomes, 23. So, here we have in total 46 number, here we have half. So, there is a reduction, and that is what we are calling it, reductional division. In other words, it is called meiotic. So, what we are trying to emphasize here regarding a terminology called spermiogenesis, where there is a differentiation of spermatids into spermatozoa. Now, once the spermatozoa is formed, this spermatozoa is kind of stick with a Sertoli cell, which we already discussed, that is also called as nurse cell, and these cells are basically giving nutrition or provide all the necessary nutrient requirement of a growing cell. So, once this spermatozoa that is already formed is traveling towards the seminiferous tubule, the process is called spermiation. Spermiation. So, spermiation is the passage of formed, fully differentiated spermatozoa towards the seminiferous tubules. Let us try to move on and start with oogenesis. Oogenesis, and this oogenesis is the formation of ova, which is a female gamete. In oogenesis, in order to understand oogenesis, let us try to understand the ovary, structure of ovary, and how the cells are developing in ovary right from the fetal life to the adulthood. Now, these cells which are available in ovary, these are precursor cells, these are mother cell actually, which is going to form or which are going to form the ovum in future, are there in the ovary right from the birth. So, during the fetal developmental stages, millions cells, the one which is going to form the mother cell is called oogonial cell. Oogonial cell. Now these oogonial cells are the mother cell. Mother cells, and these cells are formed before birth. So they are migrating in the ovary, and they are as such remain sequestered. There is no such as such growth, and also this is true that there is absolutely no addition of oogonial cell after birth. So whatever oogonial cell is present in a female body is coming right from before birth. So all the cells available in the female body is coming before birth, and after birth there is absolutely no addition of oogonial cell, and these remaining oogonial cell after birth is what is going to form the ova. Now these oogonial cells remain in the prophase 1. So these cells are basically converting into what we call primary oocyte. So primary oocytes are single cell is oocyte, and if we are talking about a multiple, we call it simply oocytes. So primary oocytes are the cells which are remain in the arrested condition. So these cells are there in the prophase first of meiotic division. So this since we are talking about again a same reductional division what we discussed in spermatogenesis. So a mother oogonial cell that which is diploid in nature. So the chromosome number is 46 here. The only difference between the male and the female here is we have 22 pairs of autosomes, and then we have a pair of sex chromosome which is XX in case of female. Now here these cells are going to give rise to a ova which is haploid in nature eventually, and that will have 23 number of chromosome. Now what we are talking here is primary oocyte that is arrested in prophase 1 of meiotic is there at the time of childbirth. Now this primary oocyte is undergoing degenerative processes, and these degenerative processes are eventually killing many of those primary oocytes, and what we are getting eventually is approximately 60,000 to 80,000 of primary oocytes. So what we have is approximately 60,000 to 80,000 of primary oocytes. So this primary oocyte after actually attaining the puberty is again converted into secondary oocyte. So basically we will get secondary oocyte, and there will be lot of developmental stages eventually, and this secondary oocyte will eventually form the mature ova, and this ova is having 23 number of chromosomes. So half the chromosome number that was earlier there in the mother cell. So this will be reductional division. So we will see that how this whole process is going one by one in terms of cellular cycle. So but bottom line is we have mother cell, oogonial cell, that is remain as such right from the birth, and then there will be no addition of oogonial cell after birth, and then this oogonial cells are converted into primary oocyte and remain as such in prophase 1 of this meiosis, and later on this primary oocyte is converted into secondary oocyte and mature ovum at the time of puberty in case of females. So we will try to understand this one in the form of a slide presentation. So if you see here we have the oogonial cell. So oogonial cells are the mother cell, and these oogonial cells are there till the fetal life. So till birth, we can simply say till birth, and after birth to puberty. So birth to puberty, there will be a stage where this primary oocyte actually will be in the, so primary oocyte will be there, and after attaining the puberty we can simply say the adult adulthood. So these primary oocyte is basically converted into these secondary oocytes, and this secondary oocyte will eventually convert into ovum, or simply we can call ova. Now these first and the second are the cells which are coming out by the meiotic division. So this actual first one is called first polar body, and the second one is called second polar body. So before actually the ova coming out from the in the female reproductive tract, what we call the process as ovulation. So ovulation is a process of getting out ova from the mature follicle. So we will try to understand the follicular biology after some time. So this second and the first polar body. So this is in diagrammatic representation of formation of ova from an Ugolian cell, and what we are getting, this polar body, is nothing but the cells coming out after the meiotic division. So this is all about, if we start looking into the left side of our panel, we have a gradation that till birth we have this kind of feature, and then after birth to puberty we have this suspended stage, and then secondary oocytes are coming during the adult age after attaining the adulthood, and then ova is released by the process of ovulation. So let's try to see the actual process of oogenesis, taking the follicle, actual follicle in example, and then we will try to understand this whole process in a great detail. So let's start and look this process in detail. So what is happening here, that we have this oogonial cell. So suppose this is a oogonial cell and ovum is here. So this is we are talking about a oogonial cell, and this is what is ovum, developing ovum. So this oogonial cell is later on converted to there will be formation of granulosa layer. So there will be a formation of granulosa cells here. So this will be granulosa cells, a granulosa layer surrounding the ovum, and this granulosa cells surrounding ovum structure in this form is what we call primary follicle. So this is what is primary oocyte. So here these are remain in the suspended position, suspended or arrested condition, what we discussed in our slide or in our previous discussion, that here the prophase one of the meiotic division is arrested. So till now they are called primary oocyte. Now this primary follicle is converted into secondary follicle by addition of more granulosa. So what will happen, eventually this cell will have more thick layer of this granulosa cell, and also addition of the thecal layer, we simply call it theca, which is nothing but the outer covering in this follicle. So this follicle, after getting some granulosa cell deposition on the top of the developing ovum and also surrounding the follicle, is what we call secondary follicle. Secondary follicle. So here we have the secondary follicle containing developing ova. Now we can correlate the whole biology in terms of the actual chromosome number and all. Now this secondary follicle, during the gradual development, what is happening, there will be a formation of cavity inside this developing follicle. Now eventually what will happen, there will be this follicle, and there will be a developing ova, and this will be surrounding granulosa cells, Actually, these are all granulosa cells, and then there will be a fluid filled cavity inside this growing follicle. So, this fluid filled cavity is called antrum. So, antrum is fluid filled cavity containing this growing ovum. Now, this fluid filled cavity having antrum and this developing ova with all granulosa cells surrounding the ova is called tertiary follicle. Tertiary follicle. Now, this tertiary follicle we can simply say is the development or progression of secondary follicle containing antrum. Antrum is nothing but the area that can have fluid material. Now, this whole tertiary follicle is matured into a very specialized follicle. So, what we are trying to discuss here, that the development of this further follicle. Now, this whole follicle will further develop into a mature follicle, and that mature follicle is having the ova that is mature, having this granulosa cell, and there will be another layer of cells which is called zona pellucida. So, we will try to delete some part in order to create some space. So, let's delete this part and see what we are talking here is the maturation of this tertiary follicle. And in tertiary follicle, what we discussed, that we have definitely this antrum, and then we have this developing ovum inside. Now, this particular additional layer of the cells surrounding the ovum is called zona pellucida. And we will see this zona pellucida will eventually form a very unique structure and a very unique area that will help the sperm to get a attachment towards the actual cytoplasm of this ova. So, once there is a penetration of a spermatozoa inside the female reproductive tract, this zona pellucida will come in contact of that sperm first, and the sperm we will see gradually the actual structure of a sperm and ova. Then we will try to analyze that how the sperm is kind of penetrating the surface of zona pellucida in order to touch this membrane and eventually to fuse the ova, the nucleus of ova. Now, this zona pellucida containing a structure which is matured is what we call Graafian follicle. Graafian follicle. So, Graafian follicle is nothing but the matured follicle having zona pellucida layer containing the ovum, and this is the structure. This structure is what is going to release this mature ova after a rupturing of this thecal layer. So, from here this is coming out, and while coming out there will be lot of fluctuation of different hormones. So, these whole process is under the control of high regulation of different hormones, which we will discuss later on. So, here this rupturing of the layer of Graafian follicle, the outer layer of the Graafian follicle will eventually release this ova or ovum out in the environment, and this whole process is called ovulation. Ovulation. So, the release of ova is by the process of ovulation, where the mature Graafian follicle is rupturing out, releasing the ova. Now, as I said, this whole process is under the control of very nice hormonal balance in the female reproductive tract. Similar thing is there in case of male reproductive system also. So, let's try to, so I'll just try to summarize here the oogonial cell, and this is exactly what is happening. What we discussed previously in terms of cellular aspect, this is just a pictorial representation of what is happening. So, this is about the oogenesis process and ovulation. let's try to see that what is the hormonal regulation of both spermatogenesis and oogenesis. So, we will cover one by one. So, first we will cover the male reproductive system and then we will cover the female reproductive system. So, let's try to see the actual hormonal or control of this gametogenesis. So, we will simply say control of gametogenesis. Control of gametogenesis. Now, so before starting this whole process, let's try to understand this whole physiological aspect of this spermatogenesis. Now, each and every mechanism, each and every process in our body is controlled by a master regulator, and that is brain. What is brain is nothing but the part of central nervous system, and central nervous system is nothing but the combination of brain and spinal cord. So, let us try to jot down one by one. What we are calling is central nervous system, and central nervous system is nothing but the collection of brain and spinal cord. Spinal cord. So, now this brain and spinal cord, we will not discuss much about the spinal cord. So, let us try to focus here in brain. What is there in the brain? Brain is a very complex organ, and many hormone, many neuronal regulation is controlled by this brain structure. In brain, there is an area called hypothalamus. Hypothalamus. And this hypothalamus is a very important area that secrete a very specific hormone called gonadotropin releasing hormone. Gonadotropin releasing hormone. So, in short, it is also called as GnRH, gonadotropin releasing hormone. Now, as name itself indicates, that it will act on gonad. So, gonado means it will act on the gonad and help gonad to release certain hormone, right. So, the whole process is gonadotropin releasing hormone. So, somehow it will go and activate gonads. Gonadotropin releasing hormone will act on pituitary, that is called master gland of our body. Now, this pituitary is what will secrete eventually two distinct hormones. One is called luteinizing hormone. Luteinizing hormone. In short, it is called LH. And second is follicular stimulating hormone. In short, it is called FSH. So, brain, the area is called hypothalamus, will secrete a hormone called gonadotropin releasing hormone, GnRH. This gonadotropin will activate pituitary to secrete two hormone, luteinizing hormone, it is called LH, and follicular stimulating hormone called FSH. Now, this luteinizing and follicular stimulating hormone will have entirely two different kind of mechanism in male as well as in female. So, we will see first male and then we will try to understand the female part. So, we will delete this part and try to focus the two hormone coming out from pituitary, and that is LH and FSH. So, we have LH and FSH. Now, this luteinizing hormone will act on an area called Leydig cell, which we already discussed. It is also called as interstitial cell, and this Leydig cell will secrete the male hormone, what we call androgens. So, androgens will be secreted from this Leydig cell, while FSH will act on the distinct Sertoli cell and other areas and help in spermatogenesis, because this will help in the formation of the follicles. So, this will act on Sertoli cell Sertoli cells in order to do our actual spermatogenesis. So this Sertoli cell will eventually help in spermatogenesis process. So this is all about the male gamete formation, the spermatogenesis process, and how LH and FSH is controlling this gamete formation in case of male. So we will try to see about female reproductive system, and to understand this female reproductive system, let's try to draw a kind of graph, and we'll see that how these two hormone, the LH luteinizing hormone and FSH, is controlling the female reproductive behavior. Now if we start looking into those oocyte, the developing follicle actually, so let's try to see here that what is happening in case of, so this is time and this is the hormone level, we can simply say the hormone level of FSH and LH. So here, so this FSH, so FSH is kind of controlling the first half of the development. So the FSH level is quite high when the follicles are developing. So follicles are developing and then FSH level is quite high while LH level is low. So LH is low and FSH is very high at the time of follicular development. At the time once the follicle maturation is done, the Graafian follicle is coming into picture, and once the Graafian follicle is formed, and if the Graafian follicle is having matured ova inside it, there is a phenomena that what we are calling it LH surge, and what is happening, this whole biology is coming upside down where the LH level is getting high. So LH is quite high and FSH is getting down, and because of this LH high level of luteinizing hormone, there is a rupture, and because of this rupture we are getting the ova coming out from that mature Graafian follicle. So this FSH is controlling the development of follicle and LH is controlling the ovulation. So LH level, LH surge, the increased level of luteinizing hormone will eventually decide the ovulation process. So these are the two distinct mechanisms, and we will try to cover it once we will discuss about the menstrual cycle in detail, that how this FSH and LH is controlled by time dependent manner. Now this is very important phenomena, and here this LH surge is controlling the ovulation. So to understand the level or the regulation of LH and FSH, let's try to look back our individual anatomical feature of the seminiferous tubule. So here what we have is interstitial cell, what we call Leydig cell, Leydig cells, and here we have a distinct cell that is called Sertoli cell, Sertoli cells. That is controlling the developing spermatozoa by providing the nourishment. Now this Leydig cell is what is going to be controlled by luteinizing hormone, and Sertoli cell is what is controlled by follicle stimulating hormone. Now this Leydig cell will eventually secrete the androgen, and this Sertoli cells will eventually help the growing spermatozoa. So let's try to understand two gametes, one from male side and the other from female side, and how this two gametes, the sperm and ova, looks anatomically, means how these specific cells are built up inside. These are not the normal cells. So first we will start with sperm, and we will see how this cell, particular reproductive gamete, what we are saying, is different than the normal human cell. And if we say the normal cell, we know that the normal cell is having just a single cell, outer is the membrane, and inside we have cytoplasm, and in cytoplasm we have lot many cellular organelle, and in between we have a nucleus, that can control the activity of this cell, right. So, what we have is lot of internal organ like mitochondria, we have lysosome, we have ribosome, we have other vacuoles and other kind of cellular organelle distributed in this cytoplasm. We have endoplasmic reticulum, we have Golgi berry and all different cellular organelles. Now, sperm is the same cell, but it is modified to do a very specific function of reproduction. Now, this cell in sperm looks like little different as compared to the one which we already discussed. Now, they have specific head. Now, this head region is having a distinct kind of shape, and this is the cap portion, and this is what you call the head region, complete head region, and this is what we have is called nucleus, and in the cap we have the structure, this is called acrosome. So, acrosome is nothing but the top portion of this head region, and then we have neck, slight depression of neck, and then we have this middle piece that is full of mitochondria. So, they are full of mitochondria, and then we have tail, and this tail is very specifically defining the motility pattern. Now, this is the same cell, but then we have in the top we have acrosome, in the head region we have nucleus and acrosome. So, nucleus is situated in this particular area, and then we have this middle piece, sometimes it is also called as body, but we will just say middle piece, and this middle piece is full of mitochondria, and mitochondria can provide energy because these are the motile cells. So, motility is coming because of this tail, and the energy to do motility is coming from this mitochondria, because mitochondria is the energy house of a cell, and this can provide energy in the form of adenosine triphosphate and what we are calling is ATP, right. So, now we have this head region, in the head region in the top we have acrosome, and if we starts looking into what are these acrosome actually. So, these acrosomes are modification of Golgi body. These are nothing but the modification of Golgi bodies or Golgi apparatus that can provide hydrolytic enzymes, and these hydrolytic enzymes are important to penetrate this sperm inside ova, and we will see this whole process at the time of fertilization. When we will discuss about the fertilization, we will see this particular area will penetrate the cytoplasm area of the ovum. So, we will see how the ova look like after discussing this. So, this is in nutshell a picture of a sperm. So, head region, middle piece and the tail region. Tail can provide the kind of movement inside reproductive tract of a female body. So, this is all about sperm, and one thing is for sure, this is all outer covering, and these are nothing but the cytoplasmic membrane. So, these are just an outer boundary similar to what we have gone in this normal cell. So, these are outer covering of the cell membrane. Now, let us start and look into ova. So, how this ova is different than the normal cell. So, there is a distinct female gamete, and that gamete is called ova. So, now we will discuss about ova. Now, ova is similar like a normal cell, and if you see, this is a normal membrane, and we are calling it as a egg membrane, or simply we can say that this is a membrane. Sometime very specifically these are called as vitelline membrane, vitelline membrane, and these are the boundary or outer surface area that can encapsulate the nucleus of ova. So, let us see, we have this as a nucleus of ova. Of course, then we will have this structure. These are nothing but the cytoplasm of the growing ova. Now, we have this structure. Now, there is a distinct area that can protect this vitelline membrane or the structure to avoid any entry of foreign cells or foreign intruder. So, we have a very specific outer covering of this ova, and this we already discussed. This is called zona pellucida. So, zona pellucida is outer covering of this ova. Now, just after zona pellucida, we have a very specific layer of cells attached to this area. So, I am not going to draw a full cell outside to this thing, but we are just drawing here. So, these cells are very specifically lining outside the zona pellucida layer, and these cells are called corona radiata. Corona radiata. Corona radiata is an outer boundary, an outer covering of this ovum. So, this is covering the outer side of the zona pellucida, and just touching this corona radiata, we have another cell layer. So, there you will see lot of protections are there, because this is such a structure. If anything is coming and rupturing this part, it's a failure in reproduction. So, this is a very important cell. So, these outer protective layer of corona radiata is called cumulus euphorus cell. Cumulus euphorus. Cumulus euphorus cell. So, these are outer covering of this ovum. So, if we try to look from outside to inside, we have cumulus euphorus cell, corona radiata, zona pellucida, white line membrane, cytoplasm and nucleus of the ovum. So, this is in total about this ova. Now, what we have to discuss little bit here, that we need to understand why this whole covering is there. Now, once there is a process of fertilization, which we will discuss in later part, the sperm is entering here. Now, the top area what we have discussed just now called acrosome is nothing but the modification of Golgi body, and that contains many enzyme, hydrolytic enzyme that can penetrate this layer, and after penetration this is covering and going and touching this area. So, we need to digest this whole area in order to travel this sperm inside this area in order to touch and ultimately cross the area to fuse with the nucleus of ovum. Now, this whole process is very important in order to do a fertilization step, and that is why this acrosome is very important, that it contains the hydrolytic enzyme. Of course, this tail and the mitochondria part in the middle piece is also important to provide motility and energy respectively in order to do a proper kind of motion inside the female reproductive tract. So, this is all about the structure details of sperm and ova.